Okay, good morning, guys. So today's lesson is going to be B3.6, which is how the digestive system works. So the learning objectives for today are to identify which enzymes break down which food group, describe how enzymes work and where each enzyme is produced, as well as which products are formed by the enzymes, and evaluate how enzymes are needed in the digestion process. Your keywords, you've got enzyme, digestion, protease, amylase, carbohydrates, lipase, fatty acids, glycerol, amino acids, proteins, and complementary. Okay, so let's go through these keywords. So enzyme. This is a tertiary or quaternary protein which is specific for its function. Enzymes are biological catalysts, remember that. Digestion. This is the breakdown of food products in the digestive tract, which is in the stomach or the small intestine. Protease is an enzyme that breaks down protein. Amylase breaks down starch, and it's usually found in salivary glands. Carbohydrates breaks down carbohydrates into disaccharides and monosaccharides. And a lipase breaks down lipids into fatty acids. And fatty acids are the monomer of lipids. Glycerol is the backbone molecule for triglycerides. Amino acid are the monomer for protein. A protein is the polymer, so it's composed of a long chain of amino acids linked by peptide bonds. And complementary is the specific shape or one type of substrate will fit an active site of an enzyme. Okay, so I'm going to give you 30 seconds just to jot down the names of each of these. So this is just a recap from way back to the first lesson. So can you name each organ of the digestive system in order? Starting with this one up here, then this one, then this one, this one, this one, and this one. So you can number them in your books and I'll know what you're on about. So 1, 2, 3, 4, 5, 6. So do 1 to 6 in your books and then write down the names of these. Around 10 more seconds. Okay, so the first one is the mouth, because that is where your food enters, and then it goes down, and this pipe here is called the esophagus. It's spelled differently in American English, so be very careful that it's actually got an O in it for us. So you've got the mouth down to the esophagus, and this extended bag is the stomach. That is where our food is churned up and broken down, or the start of the breakdown process. And then we've got the large intestine. Remember, that is the last bit that the food goes into. So we've taken all the nutrients that we need from our food that we consume, and it goes into the large intestine as a waste product. And then we've got the small intestine, which is here. So it goes from the mouth to the esophagus to the stomach, and then the small intestine, and then it goes into the large intestine. And then you've got the rectum. So that is where the waste exits the body. So also the small intestine has 3 parts to it. So you've got the duodenum, which is the first bit that the food goes into, and then you've got the ileum, and then you've got the jejunum. You don't really need to know those words. You don't need to know jejunum, but you should be aware of duodenum and the ileum. The ileum is where most absorption happens of the useful products from our food. Okay, so let's go through the pH in the digestive system. So as we know, enzymes within our bodies have an optimum temperature of 37 degrees Celsius because that is our body temperature. Remember, our body temperature ranges between 37 and 38-ish. depending on how you run as a person. Women tend to run at a warmer temperature than men. It's just how it works. Different enzymes work best at different pH levels, which we touched on last time. Proteases, for example, made in the stomach work best in acidic conditions, while proteases made in the pancreas work best in alkaline conditions. This is because the proteases that reside in the stomach have to withstand the high acidity and the pH level of the stomach, whereas ones that from the pancreas, they are secreted into the small intestine, and the small intestine is an alkaline environment. So the body makes a variety of chemicals to maintain the ideal conditions for these enzymes to work, and we will go through those now. So the glands in the stomach secrete pepsin, which is a protease enzyme. Pepsin works best at an acidic pH because the stomach is a pH 1 to 2, and the stomach produces concentrated hydrochloric acid, and we make around 3 litres a day, which is quite a lot of hydrochloric acid. As well as providing an ideal environment for proteases, it also destroys most of the bacteria consumed with food because of the high acidity and the environment of the stomach. So a thick layer of mucus protects the stomach lining from being digested by the digestive juices and the hydrochloric acid. If you didn't have this protective layer, you will essentially digest yourself because your stomach has digestive juices in it which are designed to digest protein, and your stomach is made out of protein. Essentially, you are made out of protein as a whole, so you would digest yourself if you didn't have the mucus. Okay, so when the food enters the stomach, it stimulates the secretion of hydrochloric acid from the stomach wall, and that is depicted as HCl, so hydrochloric acid is HCl. That is not an I, that is an L. Be very careful, that is hydrogen and chlorine. Okay, so the hydrochloric acid increases the acidity of the stomach to about pH 2, and that is the optimum temperature, the pH for stomach enzymes. Here we have the stomach. It is an enlarged part of the digestive system, so that's the esophagus here, and it's basically just a giant sac, which has these parts here which stop the food from escaping and the acid from escaping back up the esophagus. So we have the esophagus up here, which is the bit where the food comes down. This is the stomach itself. That's the duodenum. Remember, that's the first part of the small intestine. And in these ones here, you've got mucus cells. So remember, the stomach has to produce mucus to protect itself from the acidic environment and the enzymes. You have the gastric gland, which produces the enzymes and the mucus, as well as the hydrochloric acid, which is here. So that is the acid-producing cells, the parietal cells. So yeah, that is how your stomach is designed. So about 2 to 3 hours after a meal, the food will move out of the stomach and into the small intestine. So that'll be the duodenum that it will move into. Some enzymes which catalyze reactions are made by the pancreas and are secreted into the small intestine. So the pancreas is a leaf-shaped organ, and that has two purposes. So it has an endocrine and an exocrine purpose. Endocrine meaning hormones, so it produces hormones like insulin and glycogen, which control your sugars in your blood. And you've got the exocrine function, which is your enzymes. So it produces digestive enzymes which are secreted, not excreted, secreted into the small intestine to digest food. Other enzymes can be made by the small intestine as well, so that is important to note, and they all work best in alkaline environments. But of course that's a problem because the stomach has such a high acidity that if it just passed into the small intestine as it is, then it would burn and burn the small intestine, and it would also, because there's no mucus, and it would also denature the enzymes. So we don't need that, we don't want that. So we produce something called bile, which we will go through now. So bile is a yellow-green liquid. It's produced by the liver and it is stored in the gallbladder, and that is until it's needed. Now you can have issues with your gallbladder, which we'll get into in a moment. Bile is secreted by bile ducts. No, not ducts, ducts. We do not want ducts here. We want ducts. D-U-C-T. We do not want any ducts happening here. So, bile has a bitter taste, which you will know if you've been sick. That is, it tends to come up if you've got nothing in your stomach, and it combines with the acidic liquid in the stomach and it neutralizes it. Okay, so here we have the small intestine and the liver and the pancreas. So bile is made in the liver, which is this big organ here, and it neutralizes the acid contents coming from the stomach, creating an alkaline environment that the intestinal enzymes need to work. So here we have our pancreas, and see it's like a leaf-like structure, and remember it has exocrine and endocrine functions. Remember, exocrine means exocrine means enzymes and endocrine means hormones. And the pancreas has both these functions because it produces enzymes, which are digestive enzymes that are secreted into the duodenum, and it also produces hormones like insulin and glycagon. And if you remember, insulin means the reduction of blood sugar in the blood, and glycagon, you may not have come across before, but that is the opposite, so it increases blood sugar in the blood, which you would go into more in A level, because it's part of homeostasis and endocrine functions of the body. But basically, glycagon makes the stored glycogen, which remember is the long storage molecules for a carbohydrate such as glucose, and it will break it down and release that into the bloodstream, so you've got more in your blood. Okay, so there we go. So a little recap. So we've got the liver here, which produces bile, and then you've got the pancreas, which has two functions, so the exocrine and the endocrine functions, exocrine being enzymes, endocrine being hormones. The hormones that it produces are insulin and glycagon. Remember, insulin decreases blood sugar levels and glycagon increases blood sugar levels. And insulin is obviously the key one if you have diabetes, so you will often have insulin injections or a diet change to help. So the digestive enzymes are the ones we're going to focus on today because we are looking at digestion in the small intestine. So let's move on. So if we look at starch then, so remember starch is this long molecule made up of lots of maltose molecules. So you have amylase, which is the digestive enzyme that is symbolized by the scissors here. So that breaks up these bonds in between, which are the glycosidic bonds. So you have the starch molecule and the glycosidic bonds in between the molecules, and the enzyme, which is amylase, helps digest the starch by breaking the bonds in between the glucose molecules. So it breaks it down to maltose, which remember is two molecules joined together, so that is a disaccharide. Apologies for the appalling writing. Again, I have lost my mouse and I cannot draw with a trackpad. So the maltose is, remember, the disaccharide because it is two. So di meaning two. But it's too big to get into the small intestine. So, and the bloodstream. So it would need to fit through these gaps. So in the small intestine, the maltose is broken down into glucose units. So these bonds here are broken. So amylase is primarily found in the mouth, in the salivary glands, is made by the salivary glands, and it breaks down starch into maltose by breaking the glycosidic bond. And then when the molecules of starch get to the small intestine, they are broken down further. So they can now fit into the bloodstream. So if we go on to proteins, proteins, remember, are large branched structures. They are long chains of amino acids. So we have trypsin, which is from the pancreas, and protease from the small intestine. So trypsin from the pancreas breaks it down into slightly smaller molecules. So you'd have two amino acids joined together in they'd have peptides. So that's here. So you've got the peptides, and then the protease, sorry, from the small intestine breaks the bonds further, so you get the individual amino acids, and then they can now fit into the bloodstream. So that shows here that these are too big to fit into the bloodstream, so they need to be broken down further. Okay, so let's move on to lipids. So here you have your fats and oils, and it is in the freeze because it's a triglyceride. So bile from the liver mixes with the fats and oils to make an emulsion. An emulsion is basically a mix of products, and the lipase from the pancreas breaks down the fats to triglycerides. So the fatty acids and glycerol are the components of a fat and a fatty acid. So the enzymes have to break them down further, and you get the individual components of the fatty acid. So let's look at altering surface area. So it's important for the enzymes to have the largest possible surface area for food to work on. Fats consumed don't mix in the watery liquids in the digestive system because they are hydrophobic. So here we have some fat globules here. We have some fat globules here, and you can see that they are in water and they are sticking together, and they are staying in their shapes because they are hydrophobic. So remember, that means that they don't like water. So bile emulsifies the fats in your food, giving you lipases, giving lipases a larger surface area to work on. So that is bile's second role, which means that it breaks the fat droplets into smaller ones that the lipase enzymes can work on. Another surface which has a large surface area is the lining of the intestine, small intestine, which you would have looked at before. So remember here, you may not have seen this image, but this is a very common one for exams to use. So you've got the microvilli, and these are these little protrusions here. So you've got a very large surface area because they are basically folded up like that. So there is a large surface area, so more absorption can occur. And the small intestine, the ileum in the small intestine, is where most absorption of nutrients occurs. So not the stomach and not the duodenum, but the ileum. And I will write that down for you. Okay, so there is the ileum there. So you can see how it's written. Okay, let's go on to gallstones. So I mentioned earlier that the gallbladder can have issues. So gallstones block the bile ducts and the gallbladder, and they can range in size from a few millimeters to several centimeters. Here we have some examples. So here are some gallstones, and they reduce the efficiency of digestion because they can block the bile ducts. Not ducts, ducts, with a T, not a K. And they can be very painful. So here we can see this is a quarter. So here's a comparison of size. So they're quite big, and that can result in the gallbladder needing to be removed. And here is a 3D composite sketch of a gallbladder. So that is what it looks like. And it's quite small. So to have these large gallstones in them must be quite painful. Okay, so here is a video you need to go and watch. So this video shows you how to conduct an experiment where temperature and pH are taken into consideration against enzymes. So this is for starch, testing for starch, so remember the iodine test, and just think about some things that could be improved about this experiment while you're watching the video, and we will talk about them in a moment. So go away now, watch the video, it's in the description box, and then come back to 32 minutes and 30 seconds when you've watched the video. Okay, so you should have watched that video now. So I've written some things on the slide here. So you've got timing, volumes, and repeats, with lots of exclamation marks on that. So let's start with timing. So this is something that could have gone wrong with the experiment. If you're slightly off with your timings of getting the solution out of the test tube and then putting it in, you can't be exact every 10 seconds. So you would need a machine for that, but that could impact the results slightly. So you'll be maybe a couple of seconds out of the actual number of seconds that it took for the enzyme to change the starch into sugars. Also volumes, that is an important thing. So if you're putting in too much of, say, starch to enzyme, that's not, so it's different to what's specified in the lab report and lab notes, that could have an effect on the results. So it could take longer because there's more starch to break down. And repeats. Now this is the thing that I want to focus on. So repeats are really important in scientific practice, in scientific investigations. So you need to have a minimum of 3 repeats, because then you can identify and remove the anomalous results from your data set, which I will go through in a moment. So you need to have minimum of 3 repeats. In some experiments, you see people doing sort of 5 or 6 repeats, and that is all for the calculation of the average. So averages are important in science because you can show that you can repeat it several times and you'll get a very similar outcome. That backs up the reliability of your experiment. Okay, so minimum of 3 repeats, and let's go through anomalies. So an anomaly is a result that is not expected in that trend or data set. So it may be a lot higher in seconds than the result that you'd expect for that pH, or it might be lower than what you'd expect for that pH, for example. So doing repeats, doing 3 repeats, you can see which one the anomalous result is. So if you've got, say, a result that you'd expect 7 for, so 7 minutes, and you got 3 results which were 7, 7.2 and 5, you can clearly see that the 5 is not in the ballpark of the 7 that is expected. So the 5 would be an anomaly. I'm going to write it here so that you have the spelling. And they are fairly common in scientific experiments. So that is why we do repeats. So anomaly. Hopefully you can read that. That is an A, not an O. So that is your anomaly. So doing 3 repeats, you can clearly see if you only did 1 and you got 5 and you didn't know what the expected result is, you wouldn't know that is an anomaly. The same for if you did 2 results, you can calculate an average, but you couldn't spot the anomaly in the results in the data set. So that is just what I wanted to highlight for practicals. You will learn a lot more about this as you progress through your scientific endeavours. Okay, so here's an example of some practical data that you might have got from this practical. I am going to add some repeats on here as well so that you can calculate averages too. Okay, so I'll stop here for a moment. So on page 5, I've put 2 mock datas for repeats that you might get for pH 5. So you've got 7 here, 6.5 and another 7. For pH 5.5, there's 4.5, 4.5 and I've put 2. Now in this instance, you would probably ignore that one because it is definitely an anomalous result. Something may have got wrong in that experiment. You may have put too much of a certain solution in there, so maybe it worked quicker. You may have even used the wrong pH for it as well. So you may have mixed up tubes. So the best thing to do is to repeat it again and then see what result you get that time. So you would cross that one out and do it again. Okay, another thing that I have done here on purpose is I have done different decimal places. So if you can see here at the top, we've got to 1 decimal place. The second one is to 1 decimal place, but for the third one, for pH 6, I've actually put it to 2 decimal place for one of them. So what you would do is you would not do that. You would remove the second decimal place and just have the 1, especially if the first recording is to 1 decimal place, which it is. So keep it all the same, and then you will get the marks on the exam. So we're going to go through and do the same with the others. So cross off that one, and that is it. Oops, not meant to do that. Okay, so what I need you to do is to calculate the averages for these tests and these mock repeats. Use this one here, not the 2, because that is an anomaly, and calculate the repeats and then plot a graph for this, like you just saw in the video. This is something that you would do in the lab as well. So this is just mock data to give you an opportunity to calculate averages and also practice your graph plotting skills, which we looked at last lesson. Okay, so that's what you need to do, the averages and the graph. Okay, so here we've got breakdown protein. So this is from the book. So you would set up 3 test tubes, 1 containing pepsin, 1 containing hydrochloric acid, and 1 containing a mixture of the 2. Keep them at body temperature, so that's 37 degrees Celsius, in a water bath, and add a similar sized chunk of meat to all 3 of them. Set up a webcam and watch for a few hours to see what happens. So the conclusions you can make from this, you've got hydrochloric acid here, so that is only hydrochloric acid, there is no enzyme in there, and pepsin, and then you've got pepsin and hydrochloric acid at 37 degrees Celsius. So we can see that the hydrochloric acid on its own without pepsin it does nothing because the meat is still in there. The pepsin on its own without the hydrochloric acid, so without the acidic environment, does work slightly, but it works more slowly. So it would work eventually, but it's a really slow reaction. So that is because it is at body temperature. And then you've got the pepsin and hydrochloric acid, so that is the perfect condition for this pepsin to work because it is at 37 degrees Celsius and it has digested it fully. Okay, so we have some exam questions for you to do. These are the tasks for this lesson. So number 1, 3 types of enzymes found in the body are called amylase, protease, and lipase. Describe where each enzyme is made in the body. That one's 3 marks. Identify which reaction each enzyme catalyzes is 3 marks, and describe where each reaction works in the digestive system, and that is another 3 marks. Question 2, look at figure 1. At which pH does pepsin work best? That is 1 mark because you are just looking at these graphs here. At which pH does pancreatic amylase work best? That's also 1 mark. What happens to the activity of the enzymes as pH increases? That's 2 marks. Explain why this change in activity happens, 4 marks. And explain the importance of digestion of food in terms of molecules involved and the role of enzymes in the gut. And that's 6 marks. Okay, so you've got a total of 23 marks there. I'm not going to sit here for 23 minutes in silence. What I'm going to do is ask you to pause the video here, take as much time as you need, but 22 minutes would be the guide time, because it's 22 marks, so a minute a mark. And then resume the video and we'll go through the answers together. Again, trying very hard to write with a trackpad. So 22 guide time, that's 22 minutes. Okay, off you go. And I will go for the answers shortly. So pause the video here. Okay, so let's go for the answers. You should have all paused it and you should all have looked and attempted all of these questions. The important one that I wanted you to attempt is this bottom one here. So if you haven't done that one yet, do it now. Take 6 minutes, just bullet point some answers, and then we'll go through it. Make sure you have access to a different coloured pen as well, because that'll be easier for you to see when you go back for revision later on. So, the first one, describe where each enzyme is made in the body. So lipase is made in the pancreas, amylase is made in the salivary glands and the pancreas, and protease is made in the stomach, pancreas, and small intestine. Okay, so if you got that right, tick it. If you didn't get it right, and correct it with a different coloured pen, and then you can see where you went wrong. Okay, so identify which reaction each enzyme catalyzes. Amylase is starch to maltose, protease is protein to amino acid, and lipase is lipids into fatty acids. Describe where each reaction works in the digestive system. So amylase is in the mouth, protease is small intestine, and lipase is the small intestine as well. So look at figure 1. At which pH does pepsin work best? So find which one pepsin is on about. So you've got pancreatic amylase here, and then you've got pepsin. The optimum pH is around about here, so around about 2 or 3. So pH 3, yeah, it's around that. pH 2.5 or 3. So if you got between 2 and 3, you got it right. I'll give you that answer, that mark. And then the second one is pH 8. If you look here, go straight down and it's pH 8. At which pH does pancreatic amylase work best? That is that one. What happens to the activity of the enzymes as pH increases? So remember to link that to the structure of an enzyme. So activity decreases as the enzymes begin to denature. This is because pH affects the bonding and the structure of proteins, disrupting the bonding when pH increases. This changes the shape of the enzyme and the active site, and the enzyme can therefore no longer make enzyme substrate complexes, as the active site is no longer complementary to the substrate. So it does not work anymore. That is the basic answer. But obviously you need to write enough to get the 4 marks. So you need to say activity decreases, so that is 1 mark here. And it begins to denature. This is because pH affects the bonding and structures, so that is another mark there. This changes the shape of the enzyme and the active site, that's another mark. And that's the last mark there. So the enzyme will no longer make enzyme substrate complexes, and the active site is no longer complementary to the substrate. Okay, so the final question. So explain the importance of digestion of food in terms of molecules involved and the role of enzymes in the gut. So food needs to be broken down by enzymes into smaller molecules that can be absorbed into the gut and broken down further for resources that the body needs. Without enzymes, the food will no longer be broken down more quickly or quickly enough or at all. Therefore, the gut could not absorb the molecules and the body will be unable to survive. So Let's go through the marks. So it needs to be broken down by enzymes into smaller molecules. So that's one mark, which can be absorbed into the gut. Yep, that's another mark. And broken down further, that's another mark there. So that's 3 marks in the first sentence. Without enzymes, the food cannot be broken down quickly enough or at all. That's another mark. So that's 4 marks in total, and you justify it there. So therefore, the gut cannot absorb the molecules, and the body will be unable to survive. Okay, this is the last task that I want you to do. I want you to make your own digestive tract. So your task, should you choose to accept it, is to make a digestive tract out of household objects. Make sure you take a photo of your digestive tract and submit it along with your work on Teams. And I look forward to looking at the items that you chose for your digestive tract. You could use string or rope if you had it, and be a bit inventive. So think about what you could use to resemble the stomach, the mouth, as well as the other parts. I want you to go from the mouth down to the rectum. So do the entire digestive tract. Have fun. That should keep you busy for a while. And I will see you in next lesson. Also for next lesson, it will be a revision lesson. So I need you to, if you haven't already, note down topics that you find challenging and revise those topics, because they might be in the test. But the test will be on this topic, so it'll be digestive system and digestion. Okay, have fun.